Welcome to Crisis in Perception, where we examine the systems shaping our world.
Thank you. It's great to be here for this one.
Yeah, today's deep dive is, well, it's going to fundamentally alter how you look at a walk in the woods, or honestly, even how you look at your own behavior.
Absolutely. We're getting into some truly wild territory today.
We're exploring the book Rise of the Zombie Bugs, The Surprising Science of Parasitic Mind Control by science writer Mindy Weisberger.
And it really examines a remarkable biological phenomenon, right? Parasites that manipulate the nervous systems and the behavior of their hosts just to complete their own life cycles.
Right, because some organisms survive not by overpowering their hosts with brute force, but by quietly rewriting their behavior from the inside.
Exactly.
Which raises the big question for this deep dive. What happens when evolution discovers a way to control another creature's mind?
That is the core question we're going to unravel today. You know, when we hear the idea of zombies, we usually associate it with science fiction or horror films.
Oh, for sure. Pop culture.
Right. But Weisberger shows that forms of zombification have actually existed in nature for millions of years. Across forests, oceans, soil ecosystems, tiny organisms like fungi, worms, viruses, and parasitic wasps have evolved these incredible strategies.
Strategies that effectively turn other creatures into living delivery systems for their own reproduction.
Exactly. From a systems perspective, what we're looking at is evolutionary parasitic manipulation.
Right.
It's a biological strategy where one organism alters the behavior of another organism specifically to increase its own reproductive success.
And we have some wild, almost unbelievable examples coming up for you. I mean, we are going to look at insects being chemically forced to drown themselves.
Yes.
Spiders suddenly building bizarre architectural fortresses specifically to protect the very creatures that are killing them.
That one is fascinating.
And ants whose actual physical bodies are transformed to mimic ripe red berries. We're going to break down the what, but more importantly, we're going to spend our time today exploring the incredibly intricate how and why behind these zombie bugs.
And our mission for you today, as you listen, is to look past the superficial sci-fi and horror tropes.
Yeah, look deeper.
Right. We're going to explore the phenomenon of evolutionary parasitic manipulation as a highly rational, albeit chilling ecological network.
Because the design of this system emerges through natural selection.
Exactly. Parasites that can subtly influence host behavior gain a huge advantage if those changes help the parasite reproduce or spread.
Right.
Over evolutionary time, this pressure leads to highly specialized biological mechanisms capable of interfering with host physiology, brain chemistry, or nervous system signaling.
We'll uncover the predictable incentive structures where organisms continually adapt to influence or resist the behavior of others. Let's unpack this by starting with that word itself, zombie.
It carries a lot of baggage.
It really does. When you hear that word, your mind goes straight to Hollywood. You picture the gore-spattered ghouls from The Last of Us, or the walkers shuffling through The Walking Dead.
Or Michael Jackson's Thriller dance.
Yes, exactly. Pop culture gives us this very specific, visually terrifying image of the undead. Fictional zombies are usually mindless, battered, decaying bodies, driven by an insatiable hunger, right? They just want to bite the uninfected.
They're blunt instruments of chaos.
Blunt instruments. Perfect way to put it. But as Weisberger points out in the book, real-world zombification is the exact opposite of that.
It really is.
It is an exquisitely precise biological hack. It's a surgical intervention by an external agent that seizes control and pilots its host's mind and limbs with a very specific goal.
And that contrast is vital for understanding the underlying science here. The pop culture zombie acts chaotically. The biological zombie acts with extreme targeted purpose.
It's just that the purpose belongs to the parasite, not the host.
Precisely. The host continues to move, feed, and interact with its environment, but its behavior has been partially redirected by another life form.
Before we go deeper into the mechanics of how that redirection happens, we should probably define what we mean by zombie bugs for the context of this deep dive.
That's a good point. Entomologists would argue that the word bug only accurately refers to a specific subset of insects with sucking mouth parts, the hemipter order.
Right, the true bugs.
Yeah, but we are leaning into the colloquial, broader interpretation here. When we say bugs today, we are talking about invertebrates and arthropods.
So insects, spiders, millipedes, and even mollusks like snails.
Exactly. The mini fauna creeping around your backyard, your local parks, or the forest canopy.
So if we strip away the Hollywood fiction, how does this biological puppetry actually work? I mean, how does one tiny organism slip inside another and just take the steering wheel?
To understand the architecture of this manipulation, we have to look at a concept introduced by evolutionary biologist Richard Dawkins.
The extended phenotype.
Yes, the extended phenotype.
Mhm.
Now, if you recall from basic biology, a phenotype encompasses all the observable traits in any individual.
Right, like your hair texture or the color of a flower's petals.
Or a bird's beak shape. These are all traits shaped by a combination of genetics and the environment. But an extended phenotype refers to the expression of an organism's genes outside of its own physical body.
That's just a mind-bending concept to wrap your head around. It's like imagining a remote control car, but instead of holding a plastic controller, the driver is using raw genetic code to steer a vehicle made of living tissue.
That's a great analogy.
The parasite's genes are literally expressing themselves through the physical actions of the host.
Consider a zombified insect staggering away from its colony to die in a highly specific, seemingly random location.
Right.
That behavior isn't an accident, and it isn't a glitch in the insect's own programming. As researchers in BMC Ecology reported, that specific wandering behavior represents the parasite's genome expressing its own evolutionary needs through the body of an ant.
The insect has just been reduced to a living, breathing extension of the parasite's genetic code.
It is an extraordinary testament to the reach of natural selection.
It is deeply unsettling when you think about the implications of that. But the book makes a really crucial distinction here that we need to clarify for you before we get to the case studies.
Right, the difference between manipulation and defense.
Yeah, because not every weird, erratic behavior an infected bug exhibits is actually an example of this mind control.
Shelley Adamo, a professor at Dalhousie University who studies these neurobiological interactions, really emphasizes this. We have to differentiate between true manipulation and a host's own immune response.
And the classic example of this is behavioral fever.
Exactly, behavioral fever. If certain insects contract an infection, they might suddenly seek out an unusually warm spot.
Like a sun-baked rock or a patch of hot sand.
Yeah, and they'll just bask there for hours or days.
They are essentially trying to induce a fever to cook the parasite out of their own system. They're raising their core temperature deliberately.
That is a defensive mechanism generated by the insect's own biology. It looks like abnormal, potentially self-destructive behavior to an outside observer.
Sitting out in the open like that.
Right. But it's an evolved response that explicitly benefits the host. Similarly, a parasite might just be randomly damaging neurological tissue as it feeds.
Just chewing through wires.
Exactly. Causing the host to twitch, walk in circles, or act erratically simply because its brain is physically breaking down.
So it's just damage, not a plan.
Right. For a behavior to be classified as true parasitic manipulation, the kind of zombification we are diving into today, it must be an evolved response on the part of the parasite that explicitly benefits the parasite's reproduction or transmission.
It's not an accident. The parasite has been selected by evolution over millions of years to secrete specific compounds that modulate the host's nervous system for its own selfish gain.
That's the key.
Which brings up a question you might be asking yourself right now. How common is this? Because when you hear about these complex neurochemical mind control mechanisms, it sounds like an incredibly rare freak anomaly of nature.
Like something that only happens in one isolated cave somewhere.
Yeah, exactly. But the book reveals that it is shockingly ubiquitous.
Parasitism is arguably one of the most successful life strategies on Earth. Of the roughly 7.7 million known animal species on the planet, an estimated 40% are thought to be parasitic in some phase of their life cycle.
40%, that's almost half of all animal species.
It's massive. And specifically, the strategy of behavior manipulation has evolved independently at least 223 times across the animal family tree.
Spanning all major phyla.
Yeah, this isn't a freak occurrence. It is a highly successful, persistent evolutionary feedback loop.
Parasites that manipulate hosts more effectively reproduce more successfully, passing those traits to future generations.
At the same time, host species evolve defenses against manipulation, creating a long-term evolutionary arms race.
Let's bring that evolutionary arms race to life with our first major case study. We are moving into the fungal kingdom to look at the classic zombie ant.
The famous one.
Yeah, if you've spent any time on the science side of the internet, you've probably seen a viral video or a macro photograph of this phenomenon. It involves a fungus known as Ophiocordyceps.
This specific interaction is perhaps the most heavily studied example of an extended phenotype in action.
The behavioral override triggered by the Ophiocordyceps fungus is a masterclass in biological precision.
To really understand the scale of the manipulation, we have to look at the baseline behavior of the host, which is typically a carpenter ant.
Right. Normally, these carpenter ants spend their entire lives high up in the forest canopy. They have established foraging trails, they work cooperatively, they are highly social creatures bound to the life of their colony.
They're canopy dwellers.
Yes. But when a foraging ant is infected by an Ophiocordyceps spore that lands on its exoskeleton and enzymes its way inside, the ant's behavior radically and fundamentally shifts.
It stops acting like an ant.
Completely, it abandons its nestmates. It leaves the canopy and begins staggering drunkenly, twitching and convulsing, wandering all the way down into the understory layer of the forest, just a few feet above the soil.
And then it exhibits a highly specific behavior called summiting.
Summiting, yeah.
The ant climbs a specific piece of vegetation, usually the underside of a leaf or a prominent twig, and it clamps its mandibles down with incredible force in what biologists call a death grip.
A death grip.
And it just stays clamped there, completely immobilized, until it dies.
If we view this through the systems perspective we established earlier, we have to ask the critical question. What is the systemic benefit to the fungus? I mean, why invest the immense biological energy required to make the ant perform this exact sequence of actions?
The answer lies entirely in the parasite's reproductive needs. The canopy is too dry and hot for the fungus to develop its fruiting body.
So it can't stay up there.
No, but the forest floor is too wet, and the fungus might be washed away or outcompeted by soil microbes.
So it needs something in the middle?
It needs a Goldilocks microclimate, the precise humidity and temperature found in the understory.
Which is exactly where it drives the ant.
Furthermore, by forcing the ant to an elevated position on the underside of a leaf directly above the colony's normal foraging trails, the fungus perfectly positions itself as a biological bomber.
A biological bomber. That is a terrifying image.
It's accurate, though. After the ant dies, the fungus consumes the ant's internal organs, erupts from the back of the ant's head as a large bulbous stalk, and rains spores down onto the healthy ants walking below.
Perpetuating the infection cycle.
Exactly.
It is a flawless, self-sustaining delivery mechanism. But the how is what truly blows my mind here. Weisberger's book dives deep into the laboratory research by scientists Carissa de Becker and David Hughes.
Brilliant research.
Truly. They wanted to figure out how a mindless fungus is actually pulling the strings of a complex insect. De Becker's research found that the fungus specifically targets and alters the ant's circadian rhythms.
The internal clock.
Right. It uses highly specialized enzymes, including compounds known as enterotoxins, to essentially hack the ant's internal biological clock.
The manipulation is temporal, not just spatial. The timing of that final death bite on the leaf isn't random.
Not at all.
It almost universally occurs at solar noon, exactly midday. The fungus is dosing the ant with a cocktail of enzymes and neuromodulators to ensure the final act of clamping down happens exactly when environmental conditions, sunlight, temperature, and impending afternoon humidity are optimal for the fungus's next developmental stage.
It is adjusting the host's schedule to match its own reproductive itinerary. That level of chemical engineering is staggering.
It is. And David Hughes and his team took the investigation even further.
They did. They wanted to see the physical reality of the infection.
So they used micro-CT scans to look inside the bodies of these zombified ants at a microscopic level.
And this is where it gets crazy. Because if you imagine a parasite taking over a host's mind, you naturally assume the fungus is invading the brain, right? Wrapping around the neurons, driving the ant like a pilot in a cockpit.
That's the logical assumption.
But the CT scans revealed something completely different and honestly much more horrifying. The fungal hyphae, these thread-like networks of the fungus, actually bypass the brain entirely.
They invade the muscle tissue directly.
Yes. They weave themselves around and through the ant's individual muscle fibers in its legs and mandibles. They physically control the ant's movement like a puppeteer manipulating the strings of a marionette.
They discovered that the ant's brain is left relatively intact and untouched until the very end of the process.
Which means the ant is essentially a prisoner in its own body, just watching its limbs move without its consent.
The biological logic behind that strategy is profound, though. The fungus needs the host's brain to continue performing basic autonomous functions.
Right, it can't just kill the engine.
Exactly. The ant still needs to breathe, it still needs to maintain basic metabolic function, and it needs its navigational processing to physically walk down the tree and locate the correct leaf.
If the fungus destroyed the brain tissue too early in the infection cycle,
the ant would simply suffer a massive neurological failure and drop dead on the forest floor. The fungus would lose its transportation and never reach its elevated launch pad.
So the parasite is essentially remote controlling the muscular chassis while keeping the onboard computer running just enough to reach the destination.
It's incredible.
It really paints an incredibly vivid picture of the sheer power of natural selection. How did a fungus, which doesn't even have a nervous system of its own, learn to perform open muscle surgery on an insect?
Genetic analysis of the fungal family tree provides a really fascinating evolutionary timeline for this. Ophiocordyceps likely evolved from a much simpler, non-manipulating fungus that originally infected beetles living in the soil or inside decaying wood.
Okay.
In those early iterations, the fungus would simply infect the beetle, consume it, and kill it where it stood.
Yeah.
There was no need for complex mind control because the beetle was already in a suitable environment for the fungus to reproduce.
But then something changed.
Right. At some point in deep evolutionary history, the fungus made a massive ecological leap. It jumped to parasitizing social insects,
Yeah.
specifically ants.
And as the book details, trying to parasitize an ant colony is a completely different ballgame than attacking a solitary beetle in the dirt.
Ants possess a highly sophisticated defense mechanism known as robust social immunity.
They operate as a superorganism.
Yes. If a single ant in a colony becomes infected with a pathogen, the other worker ants do not nurse it back to health.
No bedside manner.
None. They actively police the colony. They will detect the chemical signature of the illness, isolate the sick ant, kill it if necessary, and physically carry the infected body far away from the nest to a designated waste site.
They do this to protect the dense, vulnerable population of the colony.
So when the ancestral fungus jumped to ants, it faced an insurmountable systemic barrier. If it simply killed the ant inside the nest, the colony's sanitation department would immediately dispose of the threat.
Permanently breaking the parasite's transmission cycle. So to survive in this new, hostile environment, the fungus had to evolve a workaround.
It had to invent a mechanism to force the ant to leave the safety of the colony of its own volition before the infection became obvious.
And before the ant died.
Exactly. The entire phenomenon of the zombie ant, the staggering, the summiting, the death grip, was forged by the evolutionary pressure of simply trying to bypass an ant colony's garbage disposal system.
That's wild. The evolutionary pressure to evade social immunity forged this incredibly complex behavioral manipulation.
However, systems analysis shows us that extreme specialization always comes with a trade-off.
Right, nothing is free.
To control an ant's behavior with such surgical precision, to perfectly hack its specific circadian rhythms, to weave through its specific muscle structures without triggering an immediate fatal immune response, the fungus had to become hyper-specialized.
It is so tightly co-evolved with one specific species of host that it loses the ability to be a generalist.
An Ophiocordyceps strain that successfully zombifies one specific species of carpenter ant in the Amazon often cannot infect a different, closely related species of ant walking on the exact same tree.
It's the ultimate evolutionary gamble. The fungus becomes the undisputed master of one very specific domain, but it is entirely locked into the survival of that single host species. If that ant goes extinct, the master manipulator goes down with the ship.
This high-stakes specialization isn't limited to fungi that attack ants, though. The evolutionary pathway of fungal mind control has emerged in other insect orders, utilizing completely different mechanisms to achieve a similar goal.
That brings us to a perfect contrast. The zombie flies. While the ants are fascinating, the research on flies infected by a fungus called Entomothera muscae introduces a whole new level of chemical manipulation.
Entomothera muscae shares the overarching strategy of inducing summiting behavior, compelling the host to seek an elevated position to disperse spores.
Right.
But the temporal trigger and the internal mechanics are distinctly its own.
Where the zombie ant is driven by its hacked circadian rhythm to bite down exactly at midday, this fly fungus operates on a different schedule.
It forces the infected housefly or fruit fly to summit precisely at sunset.
Sunset. As the sun goes down, the fly climbs up a wall or a blade of grass, extends its wings outward and upward in this rigid, bizarre pose designed to fully expose its abdomen, and it dies.
And then the fungus forcefully ejects spores from the fly's exposed abdomen into the cooler evening air.
But the real mind-bender here is the research by scientist Carolyn Elia on how the fungus drives the fly upward. It is like a biological horror story.
Elia's work is a masterclass in isolating the variables of parasitic manipulation. She designed a study to determine whether the manipulation was localized in the nervous system or circulating systemically throughout the fly's body.
She performed this procedure where she extracted hemolymph, which is the insect equivalent of blood.
Right.
She took it from an infected fly that was right in the middle of its frantic zombie summiting phase.
Peak zombification.
Yeah. She then took that infected, chemically loaded bug blood and transfused it directly into a completely healthy, uninfected fly.
And the results were instantaneous.
Almost immediately after receiving the transfusion, the healthy fly exhibited accelerated locomotion. It started behaving erratically, pacing, and moving upward, mimicking the zombie behavior just from the introduction of the infected fluid.
The implications of that transfusion experiment are profound. It proves that the behavioral override isn't just a physical consequence of the fungus growing inside the fly's body cavity.
It's active.
The fungus is actively synthesizing and releasing highly potent behavior-altering molecules directly into the host's circulatory system. Elia appropriately referred to these fungal parasites as natural neuroscientists.
Natural neuroscientists.
The fungus appears to possess the ability to weaken the host's blood-brain barrier,
Wow.
allowing it to pump these specific chemical compounds directly into the fly's central nervous system to manipulate neural activity. It is manufacturing a custom pharmaceutical intervention to seize control of the fly's brain.
It is chemically hacking the brain to force a fatal sunset climb. And speaking of hacking, fungi are far from the only organisms that have mastered this art.
Not at all.
The book transitions into an entirely different biological kingdom, examining insects that manipulate other insects. We're going to look at wasps and viruses next, and the sheer cruelty of the evolutionary strategies at play here is staggering.
Truly brutal.
The author introduces us to the work of parasitologist Kelly Weinersmith, who studies what I can only describe as a biological matryoshka doll of horrors. She researches a specific insect called the crypt keeper wasp.
Uterus set, the crypt keeper wasp, belongs to a category known as parasitoids.
Parasitoids.
Unlike a standard parasite, which often benefits from keeping its host alive for as long as possible to continuously extract resources, a parasitoid's life cycle inevitably results in the death of its host.
The host is merely a temporary developmental chamber and a food source.
To understand the crypt keeper, you first have to understand its victim, the gall wasp.
Gall wasps are fascinating in their own right. They lay their tiny eggs into the living tissue of a tree, often an oak tree.
And the tree reacts to this intrusion by forming a tough, woody, protective growth called a gall.
Entirely around the developing wasp larva. It's essentially a custom-built, fortified nursery provided by the tree.
Normally, when the gall wasp larva matures into an adult, it uses its powerful mandibles to chew a nice, large, comfortable hole through the woody plant tissue, crawls out, and flies away to start the cycle again.
Peaceful process.
But the crypt keeper wasp intercepts this peaceful process. The female crypt keeper uses a long, needle-like ovipositor to pierce through the woody exterior of the gall and inject her own egg directly into the sealed chamber containing the developing gall wasp larva.
So now there's two of them in there.
The crypt keeper egg hatches, but the resulting larva doesn't immediately consume its host. It waits.
Because it needs a way to escape the tough, woody gall once it reaches adulthood.
And its own jaw structure is significantly weaker than the gall wasp's. It lacks the physical tools to chew its own way to freedom.
So the parasite essentially outsources the manual labor. It forces the host to do the heavy lifting. As the gall wasp prepares to emerge, the crypt keeper larva manipulates the host's behavior.
But here is the truly diabolical part. The parasite chemically forces the gall wasp to alter its normal chewing pattern.
Instead of chewing a large escape hatch, the host is compelled to chew a hole that is deliberately too small for its own body to fit through.
It's agonizing to think about.
The gall wasp chews this inadequate hole, tries to squeeze its body out to escape, and predictably gets its head permanently wedged in the opening. It is trapped, plugging the hole completely like a cork in a wine bottle.
This agonizing scenario serves two vital systemic functions for the parasitic crypt keeper. First, by using the host's stuck body to plug the hole, the parasite effectively seals the entrance against external predators or secondary parasites that might try to enter the gall while the crypt keeper finishes its final stages of maturation.
It turns the host into a living doorstop.
Second, it provides the perfect custom-sized escape hatch. Once the crypt keeper larva is fully developed, it consumes the internal organs of the trapped, immobilized host from the inside out. When it is ready to emerge into the world, it literally crawls out through the host's empty, hollowed-out head capsule.
That is just rough. Kelly Weinersmith actually proved how crucial this horrifying manipulation is to the parasite's survival.
She did.
Her team conducted an experiment where they went into the field and glued tiny, thin pieces of bark over the partially chewed crypt openings, trapping both the host and the parasite inside.
And they found that if the adult crypt keeper was forced to try and punch through even a thin layer of bark on its own, it was 3 times more likely to die trapped inside the gall.
It absolutely relies on the host's labor, and it relies on perfectly timing the death of its host so the hole is exactly the wrong size. It's a chilling reminder that natural selection has no concept of mercy or ethics.
It only rewards what works. The precision required to manipulate that specific hole-chewing behavior, likely by tinkering with the host's hormonal development or neural pathways, showcases the astonishing depth of this evolutionary arms race.
And this manipulation of host labor extends to protection as well. Let's look at another horrifying wasp example from the book. Did you know that parasitic wasp is actually served as the direct inspiration for the legendary sci-fi horror movie Alien?
That's a great piece of trivia.
Weisberger mentions this fantastic anecdote about director James Cameron. He had this intense, visceral fever dream about being trapped in a room completely covered in crawling masses of wasps.
Terrifying.
He was terrified to move, paralyzed by the sheer biological wrongness of it. He took that feeling of absolute paralyzing dread and the concept of an organism using another creature as an incubator and translated it into the famous xenomorph chest burster sequence.
And honestly, the reality of what parasitic wasps do to caterpillars is arguably worse than the movie.
It really might be.
We're looking at the phenomenon of the remote control caterpillar bodyguard. Several species of bracketed wasps utilize caterpillars as living incubators and subsequently as fiercely aggressive sentinels.
The life cycle starts when a female wasp lands on a caterpillar and rapidly injects dozens of microscopic eggs directly into the caterpillar's bloodstream.
The caterpillar survives the initial attack and goes back to munching on leaves.
Meanwhile, the wasp eggs hatch inside. The larvae begin feeding on the caterpillar's non-essential fluids, carefully avoiding the vital organs so the host stays alive and continues to eat and grow.
Inadvertently gathering nutrients for the parasites inside it.
Eventually, the dozens of wasp larvae reach maturity. They release chemicals that temporarily paralyze the caterpillar, and then they literally chew their way out through the caterpillar's skin simultaneously.
There's a mass exodus.
Once outside, they immediately spin these little protective silk cocoons right next to the bleeding caterpillar to begin their pupal stage. Now, common sense would suggest that a caterpillar that just had dozens of creatures chew through its exoskeleton would either immediately die from the trauma or at the very least crawl away as fast as possible to try and heal.
Under normal biological circumstances, those survival instincts would kick in. But under the influence of parasitic manipulation, the normal software is completely overwritten.
The injured caterpillar does not leave the scene.
Instead, it alters its behavior to become a dedicated protector. It spins a protective silken shield over the cluster of wasp cocoons. It stops eating entirely. Its own biological imperative to metamorphose into a moth or butterfly is permanently suspended, and it transitions into a state of hypervigilance.
Researchers set up an experiment to test just how aggressive these zombified bodyguards become.
Right, the paintbrush test.
They approached the cocoons and gently poked the guarding caterpillar with a tiny, soft paintbrush to simulate the approach of a predator, like a stink bug or a spider. The caterpillars absolutely lost their minds.
They thrashed the front half of their bodies violently back and forth.
They regurgitated toxic fluids, and they aggressively bit at the bristles of the paintbrush. They actively, violently guarded the very creatures that just spent weeks eating them from the inside out.
And they maintained this frantic defensive posture until the adult wasps finally hatch from the cocoons and fly away. Only then does the exhausted, starving caterpillar finally succumb to its injuries and die.
The true crisis in perception here, the element that completely upends our traditional understanding of biology and interaction, is the mechanism of control. Think about the timeline.
Yes, the timing is the key mystery.
When the caterpillar is exhibiting this violent, dedicated guarding behavior, the wasp larvae are already outside of its body, sealed away inside their own inert cocoons.
They are in a pupal state. They aren't feeding, and they certainly aren't physically interacting with the caterpillar's nervous system anymore.
So how is the wasp continuing to manipulate the host's brain remotely?
The answer involves a third party. Enter the polyviruses, or PDVs. This is where the story shifts from a simple predator-prey relationship to a bizarre biological partnership.
It is a remarkable example of mutualism functioning within parasitism.
Millions of years ago, an ancestor of these wasps incorporated a virus into its own genome. Over vast evolutionary timescales, this virus lost the ability to replicate and exist on its own in the environment.
It now exists entirely in an obligate symbiotic partnership with the wasp. It's essentially a biological weapon that the wasp manufactures and carries in its arsenal.
Exactly. When the mother wasp injects her eggs into the caterpillar, she simultaneously injects a massive dose of this polyvirus. The virus immediately goes to work on two fronts.
What's the first front?
Its first evolutionary purpose is to suppress the caterpillar's highly sensitive immune system, so the host's white blood cells don't encapsulate and destroy the foreign wasp eggs.
Okay, that makes sense.
Its second purpose is neurological. The viral particles enter the caterpillar's brain and begin altering the host's hormones. The virus physically rewires the caterpillar's neural pathways to stop it from molting, to suppress its appetite, and ultimately to force it into that hyper-aggressive guarding stance once the larvae exit.
So the virus does the neurological hacking on behalf of the wasp long after the wasp larvae have left the host's body.
The manipulation is a joint venture between an insect and a virus.
A joint venture between a wasp and a virus to turn a caterpillar into a zombie bouncer. The complexity of these systems is just staggering. Let's look at one more master manipulator in the wasp family, focusing on architectural sabotage.
The spiders.
Yes. The book details the work of researcher William Eberhard in Costa Rica, looking at how wasps manipulate the engineering skills of spiders.
Eberhard's research focuses on the Zatopoda wasp and its interaction with the Anelosamus spider. This represents a shift from manipulating simple locomotion or aggression to hijacking highly complex, innate construction behaviors.
We all know that spiders are master builders. They possess the innate knowledge to build intricate, geometrically perfect, sticky orb webs designed specifically to catch flying insects for food.
But certain parasitic wasps look at a spider and see a free contractor.
The interaction begins when a female wasp attacks a spider in its web. She delivers a highly precise surgical sting directly into the spider's main nerve ganglion in its brain to temporarily paralyze it.
Just a temporary knockout.
While the spider is immobilized, the wasp lays a single tiny egg on the spider's abdomen, right near where the spider cannot reach to groom it off.
And then the spider eventually wakes up from the paralysis, shakes itself off, and goes completely back to normal.
For a week or two, it repairs its orb web, catches flies, and acts like nothing happened. Entirely unaware that the wasp larva hatched and is now clinging to its abdomen, slowly piercing its exoskeleton and drinking its hemolymph.
The host is kept healthy and productive, unwittingly sustaining the parasite. But as the wasp larva reaches the end of its developmental cycle, it initiates a profound and sudden behavioral shift in the spider.
The larva needs a safe, secure place to pupate, and a delicate, sticky orb web exposed to the elements and predators is insufficient.
So, the night before the wasp larva is ready to pupate, it injects a chemical cocktail into the spider that completely overwrites the spider's web-building software.
The spider abandons its normal, intricate design. Instead, it spends hours obsessively building a highly specific, drastically simplified, and heavily reinforced structure known as a cocoon web.
It stops using sticky silk entirely. Instead, it lays down thick, durable structural threads, reinforcing the same lines over and over again to create a taut, sturdy, tent-like structure.
This new web is designed specifically to be incredibly durable, to withstand high winds, and to efficiently shed tropical rainwater.
Once this architectural masterpiece is finished, the spider settles into the dead center of the fortress it just built.
The wasp larva then delivers a final, fatal dose of venom, drains the last fluids from the spider's body, discards the empty husk, and spins its own pupal cocoon safely suspended in the middle of this custom-built, heavily fortified nursery.
The specificity of the neurochemical hack is astounding. The wasp venom and subsequent chemical injections don't just scramble the spider's brain into building random, chaotic tangles of silk.
It's not just breaking things.
No, they activate a highly specific, normally dormant subroutine in the spider's nervous system. Spiders occasionally build similar, reinforced resting webs when they are preparing to molt, a time when they are highly vulnerable.
The wasp parasite has evolved the precise chemical key to unlock and hijack that specific survival behavior, redirecting the spider's innate architectural abilities entirely for its own protection.
The parasite finds the blueprint for a bunker hidden in the spider's brain and forces the spider to build it.
That transitions us beautifully into the next major category of our deep dive. We've seen fungi and wasps. Now let's look at worms and flies. The book features some examples that I can only describe as brain invaders and body snatchers.
And we have to start with the decapitating flies, because the methodology here is a masterclass in deception.
We are looking at Pseudaction tricuspis, commonly known as the phorid fly. Their interaction with invasive fire ants is a brilliant example of what ecologists call a slow con.
The entire strategy is built around latency and delayed gratification.
The mother phorid fly hovers over a foraging fire ant and, in a fraction of a second, dive bombs the ant and uses a specialized ovipositor to inject a tiny egg into the ant's thorax.
The egg hatches into a microscopic maggot, which then slowly navigates through the ant's internal fluids and migrates all the way up into the ant's head capsule.
And then it just stops. It sits there.
For weeks, the maggot lives comfortably inside the ant's head, sipping on the ant's bodily fluids, slowly growing larger and larger, essentially taking up residence right next to the ant's brain.
And during this entire multi-week period, the ant goes about its daily business as if nothing is wrong. It forages for food, it interacts with its nestmates, it participates in colony defense, showing absolutely zero external signs of infection or distress.
This extended period of latency is the crucial element of the parasite strategy. As we discussed earlier with the Ophiocordyceps fungus, ant colonies possess a ruthless system of social immunity.
Right. If the infected ant showed any sign of illness, lethargy, or erratic behavior, its nestmates would immediately execute it and remove the body, destroying the growing maggot.
By remaining entirely passive and chemically invisible, allowing the host to behave perfectly normally, the fly maggot evades the colony's sophisticated defenses while it safely utilizes the host's resources to mature.
It's the ultimate sleeper agent. But eventually, the maggot runs out of room in the ant's head, and the switch flips.
Just 8 to 10 hours before the maggot is ready to enter its pupal stage, it finally activates the zombification sequence.
The ant abruptly stops whatever it's doing, abandons its normal duties, and leaves the safety of the nest. It wanders out alone into the thatch layer of the soil, seeking a secluded, moist microenvironment away from the colony's foraging paths.
It departs on a one-way expedition, guided by the chemical cues released by the maggot. Once the ant arrives in a location deemed suitable by the parasite, the endgame begins.
The maggot releases a specific blend of enzymes that actively dissolve the connective tissue and membranes, holding the ant's head to its thorax.
The ant's head simply falls clean off its body.
That's off.
The maggot then rapidly consumes whatever remaining tissue and brain matter is left inside the severed head, pushes the ant's mouthparts out of the way, and pupates right there inside the empty head capsule.
Using the hard, chitinous shell of the ant's face as a protective bunker against predators while it transforms into an adult fly.
The efficiency of the system is remarkable. The parasite uses the host for long-term nutrition, for transportation to a safe, highly specific pupation site, and finally repurposes the host's own exoskeleton as armored housing.
Moving from decapitation, let's explore a parasite that utilizes extreme body modification. This is where zombification goes beyond just altering a host's behavior and actually physically restructures the host's appearance to trick a third species.
The book outlines the incredible research of Steve Yanoviak concerning a nematode worm and what is affectionately known as the berry butt ant.
Berry butt ant?
Yes. This complex, multi-species interaction involves a nematode worm called Myrmecanema neotropicum and its host, the common giant turtle ant, which inhabits the high canopies of the South and Central American rainforests.
Normally, these giant turtle ants are completely black from head to toe. They scurry around the canopy looking for food, but when an ant becomes infected by this specific nematode, the worms migrate toward the ant's gaster.
The large, bulbous rear segment of its abdomen.
Right. As the worms rapidly reproduce and mature inside the gaster, they cause the ant's exoskeleton to thin out and physically swell up. Simultaneously, they alter the pigmentation of the ant, changing the gaster from black to a bright, translucent, vibrant red.
The ant's rear end is physically transformed to perfectly mimic the appearance of a small, ripe, juicy forest berry.
To understand why a worm would turn an ant into a fake berry, we have to look at the closed loop of the biological system. The nematode's definitive host, the environment where it must ultimately end up to sexually reproduce and complete its life cycle, isn't an ant at all.
It is a frugivorous or fruit-eating bird.
The nematode needs to get inside a bird, but birds don't generally eat spicy, hard-shelled ants. They eat fruit. So the nematode disguises the ant as a piece of fruit.
But a physical disguise isn't enough to complete the con. The ant has to act like a berry, too.
Because if the red ant just sprinted around the branches or hid under leaves like it normally does, the bird wouldn't be fooled.
Exactly. So the parasite pairs the physical modification with behavioral mind control. It alters the ant's nervous system, making it incredibly sluggish.
It forces the ant to abandon its evasive instincts and spend an inordinate amount of time foraging out in the open, exposed on the tips of branches, actively holding its bright red rear end elevated in the air.
A hungry bird flies by, spots what it thinks is a stationary, juicy red berry hanging on a branch, and snaps up the ant.
The bird digests the ant, but the nematodes survive the digestive process. They safely reproduce inside the bird's intestinal tract. As the bird flies continuously through the vast forest canopy, it defecates.
And the bird droppings, now heavily loaded with thousands of microscopic nematode eggs, land on the leaves below.
Now, how do the eggs get back into the ants? Worker ants regularly forage for bird droppings to use as a protein-rich food source for their growing larvae back in the nest.
The workers collect the infected poop, carry it back into the colony, and feed it directly to the vulnerable ant larva.
The larvae ingest the eggs, the nematodes hatch, the larvae mature into infected adults, their gasters turn red, they walk out onto the branch, and the entire multi-species cycle seamlessly restarts.
It is a breathtakingly complex, perfectly closed loop of ecological manipulation. The parasite successfully bridges the gap between an insect and an avian predator by rewriting the physical body and the behavioral instincts of its host.
And speaking of parasites that make their hosts look like a tasty snack, we have to mention the disco-eyed snail.
Leucochloridium, a parasitic flatworm that employs a highly visual, incredibly dramatic strategy.
If you search for videos of this online, it is equal parts mesmerizing and horrifying. This parasitic worm infects a common land snail and migrates into the snail's translucent eye stalks.
The worms form these massive, swollen, brightly colored bands of green, yellow, and red inside the eye stalks.
They actively pulsate and wriggle in a rhythmic motion that perfectly replicates the appearance of a fat, juicy caterpillar crawling on a leaf.
But again, the physical disguise is useless without the behavioral hack. Snails are naturally photophobic. They hate the sun.
Their survival depends on hiding in the dark, damp undergrowth during the day to avoid drying out and to hide from visual predators.
But the leucochloridium parasite, much like the nematode we just discussed, needs to be eaten by a bird to reach its definitive host stage in the bird's digestive tract. A bird is never going to find a snail hiding deep under a log.
So the parasite chemically overrides the snail's deep-seated, innate aversion to light. It reverses the snail's instincts.
It forces the snail to creep out of the safe shadows and systematically climb high up onto exposed, sunlit leaves in broad daylight.
There, entirely exposed, the snail vigorously pulses its fake caterpillar eye stalks to physically flag down passing hungry birds.
It's essentially forcing the host to hold up a flashing neon sign that says, "Eat me."
It is driving the host straight into the mouth of a predator. From parasites that force their hosts to seek the light, we move to a parasite that forces its host to seek the water. This brings us to the suicidal mantis.
This manipulation involves a type of nematode known as a hairworm, specifically a species from the genus Chordodes, which specializes in infecting predatory insects like praying mantids.
The setup here is incredibly specific. A tiny hairworm larva is ingested by a praying mantis, likely through a secondary host like a mosquito that the mantis ate.
The hairworm takes up residence inside the body cavity of the mantis, absorbing nutrients and growing rapidly. These worms can grow to astonishing lengths, sometimes growing significantly longer than the mantis itself, coiling up tightly inside the host's abdomen.
But the host mantis continues to hunt and live normally. But here's the biological conflict. The adult hairworm is an aquatic organism.
It absolutely must return to a body of water, a pond, a stream, or even a puddle to mate and lay its eggs.
But a praying mantis is a strictly terrestrial insect. It has no business being near water, and it certainly doesn't swim. So how does a massive worm trapped inside a dryland insect get to a pond?
It achieves this by profoundly altering the host's visual perception, effectively driving the mantis toward aquatic environments.
The book highlights a brilliant experiment conducted by researchers in Japan to prove exactly how this visual hacking works. They wanted to know if the worm was just making the mantis randomly erratic or if it was specifically targeting the insect's vision.
They set up a mesh enclosure and placed infected mantids inside, At one end of the enclosure, they placed two artificial pools of water.
One pool was designed to reflect dim light that was strongly horizontally polarized.
Horizontally polarized light is the exact, specific optical signature that occurs when natural sunlight reflects off the flat surface of a pond or a lake.
The second pool reflected a much brighter light, but it was weakly polarized, meaning it just looked like a bright, shiny surface, not necessarily a deep body of water.
The results of the behavioral trial were highly definitive. The infected mantids ignored the brighter light and overwhelmingly chose to walk directly toward and ultimately hurl themselves into the pool reflecting the dimly lit, horizontally polarized light.
The parasite didn't just make the mantis thirsty, it actively hijacked the insect's visual processing center. It lowered the mantis's threshold for detecting horizontally polarized light, forcing the insect to be inexplicably magnetically drawn to the specific optical signature of water.
The mantis sees the shimmer of the water, marches to the edge, hurls itself in, and immediately begins drowning.
And the moment the mantis hits the water and starts to die, the massive hairworm violently bursts out of the host's rear end, uncoiling itself into the water to swim away and find a mate, leaving the hollowed-out mantis to sink.
The researchers took this a step further and actually analyzed the specific proteins the parasite produces inside the host's brain during this final manipulation phase.
They found novel proteins linked directly to the release of neurotransmitters, effectively hot-wiring the brain. But even more shockingly, they found proteins connected to cellular apoptosis.
Apoptosis is the biological mechanism for programmed cell death. It's a cell's self-destruct sequence.
The presence of those specific proteins suggests a profound and aggressive level of physiological control. The worm isn't just tricking the brain, it may be causing specific cells in the host's central nervous system to literally commit suicide, selectively wiping out resistance pathways to ensure the mantis doesn't fight the urge to jump into the water.
It's like a computer hacker not just taking over your machine, but actively deleting your antivirus software and wiping your hard drive so you can't reboot. It is absolute biological domination.
And hearing about these complex neurological overrides raises a deeply uncomfortable question, a question that moves us into the final section of our deep dive today: mammals, humans, and the illusion of control.
This is where it gets personal.
It really does. We've spent this entire time talking about ants, flies, wasps, spiders, and snails, and it is very easy to listen to this and think, well, they are just bugs. They have simple, rudimentary brains. Of course, a fungus or a worm can hack them. That could never happen to a complex mammal like us.
What could it?
Exactly.
It is a natural defense mechanism to assume human exceptionalism, but biology paints a different picture. Humans have a deep, ancient, and unbroken history of co-evolution with parasites.
The text highlights archaeological evidence that underscores this intimacy. Researchers have discovered the eggs of parasitic protozoans perfectly preserved in biblical-era toilets excavated in Jerusalem.
They have found the remains of severe intestinal parasites in mummies dating back to China's Ming Dynasty.
And they have cataloged Bronze Age coprolites, which are fossilized human feces found in ancient English settlements that are absolutely loaded with diverse parasite eggs.
Evolutionary biologists categorize human parasites into two groups: heirloom parasites that we inherited from our primate ancestors and carried with us out of Africa, and souvenir parasites that we acquired from other animals as we domesticated livestock and spread across the globe.
We are walking ecosystems, and we have always been carrying passengers. And yes, certain microbial zombifiers have very well-established track records for completely altering the behavior of large, complex mammals.
The most famous example, which we often take for granted, is the rabies virus.
Rabies.
Rabies, caused by the lysovirus, is a stark example of a pathogen weaponizing mammalian biology for its own transmission. The virus enters through a bite and slowly travels along the peripheral nerves until it reaches the central nervous system. Once it infiltrates the mammalian brain, it causes severe targeted inflammation.
But it doesn't just destroy brain tissue indiscriminately, turning the host into a vegetable. That wouldn't help the virus spread.
Exactly the opposite. The inflammation specifically targets the limbic system, the area of the brain that regulates emotion and behavior. The virus suppresses the host's rational fear responses and heavily promotes extreme, unprovoked aggressive behavior.
It simultaneously paralyzes the throat muscles, making it impossible to swallow, which causes infected saliva to pool in the mouth.
The sequence perfectly incentivizes the host, whether it's a domestic dog, a wild raccoon, a bat, or a human, to violently bite other animals.
Because the virus concentrates itself in those salivary glands, that highly aggressive bite is the perfect engineered transmission vehicle to infect a new host. The aggressive, rabid behavior is a classic extended phenotype of the rabies virus manifesting in a mammal.
But rabies is visually obvious. If you see a foaming, highly aggressive raccoon, you know what's happening. The symptoms are loud.
Let's talk about a parasite whose symptoms are incredibly quiet. Let's look at what might be the ultimate stealth mine hacker, toxoplasmosis, caused by the single-celled protozoan Toxoplasma gondii.
Toxoplasma gondii is perhaps the most fascinating case study in subtle, systemic behavioral manipulation in mammals. Its life cycle revolves around a highly specific, definitive host, the feline.
T. gondii can only sexually reproduce inside the intestinal tract of a cat. But the parasite needs a way to get into the cat. The cat sheds millions of microscopic T. gondii oocysts, or eggs, in its feces.
Those eggs contaminate the surrounding soil and water, where they can be inadvertently ingested by almost any warm-blooded animal foraging in the area.
The classic intermediate host in this evolutionary drama is a rodent, like a mouse or a rat. Now, under normal circumstances, a mouse is biologically hardwired to be utterly terrified of cats. The mere scent of cat urine is enough to trigger a massive stress response and send a healthy mouse running in the opposite direction.
It is a fundamental survival instinct deeply embedded in the rodent strain. However, when a rodent ingests T. gondii eggs, the parasite navigates to the rodent's brain and forms microscopic cysts within the neural tissue, particularly heavily concentrated in regions like the amygdala, which processes fear and emotion.
The presence of these cysts effectively performs a targeted chemical lobotomy on the rodent's fear center. The infected rodent permanently loses its innate fear of cat urine.
In fact, studies show that instead of fleeing, the infected mouse becomes mildly attracted to the scent.
Oh.
It will actively explore areas marked by cats, significantly increasing the probability that it will be intercepted, caught, and eaten by a feline predator. By altering the host's perception of risk, the parasite successfully returns itself to the cat's intestines to reproduce.
It serves the mouse up on a silver platter by simply turning off its alarm system. But here is where this deep dive gets incredibly relevant for you listening right now. Humans are warm-blooded mammals. We are just as susceptible to ingesting T. gondii as a mouse is.
We can be infected by eating undercooked meat from livestock that grazed on contaminated soil, by eating unwashed garden vegetables, or, yes, by accidentally inhaling particles while cleaning a domestic cat's litter box.
And the prevalence of this parasite is staggering. It is estimated that billions of human beings, roughly 1/3 of the entire global population, currently carry latent T. gondii cysts in their brains.
For a very long time in medical science, it was assumed that these latent infections in humans were entirely asymptomatic. The consensus was that unless an individual had a severely compromised immune system, the cysts just sat there, dormant and harmless, heavily suppressed by the human immune response.
However, emerging research in the field of neuroparasitology suggests a much more complex and subtle ongoing interaction with the human brain. The book highlights a truly fascinating piece of research conducted by Stephanie K. Johnson and Peter Johnson at the University of Colorado Boulder.
They decided to look at the potential macro-level effects of this parasite on human society.
They gathered extensive business and economic data from 42 different countries around the world, measuring the rate of entrepreneurial activity. They then cross-referenced that economic data with the known prevalence of T. gondii infections in the general public of those same 42 countries.
And the results they found showed a startling correlation. Countries with higher baseline infection rates of the parasite consistently demonstrated significantly higher rates of entrepreneurship and small business creation.
On the surface, correlating a cat parasite with starting a business seems entirely counterintuitive. But when you break down the underlying neurochemistry, a distinct systemic logic emerges. Entrepreneurship, by its very nature, involves a high degree of risk tolerance.
It requires an individual to overcome the natural, rational fear of failure, financial ruin, and social stigma.
The researchers hypothesized that the microscopic cysts formed by T. gondii on the human brain trigger localized, subtle immune responses. These low-level inflammatory responses can disrupt the normal production and communication pathways of key neurotransmitters, specifically dopamine and serotonin, the chemicals largely responsible for regulating stress, anxiety, and fear assessment.
Think about the parallel here. The parasite reduces the mouse's rational fear of the cat, so it takes a fatal risk. In a human brain, the parasite's presence might be subtly reducing our rational fear of taking a massive professional or financial risk.
Stephanie Johnson pointed out that T. Gandhi isn't necessarily making people smarter or better at business. It might just be taking the edge off that paralyzing fear of failure, leading to slightly more risk-taking behavior across an infected population.
The implications of that hypothesis are profound. We're talking about a microscopic, single-celled parasite whose sole evolutionary goal is simply to find its way back inside the digestive tract of a feline.
Yet, as an accidental byproduct of its residency in the human brain, it subtly rewires human neurochemistry just enough to potentially influence macroeconomic behaviors, risk assessment, and global business trends.
It demonstrates how deeply intertwined our seemingly autonomous decisions are with the unseen biological world around us.
It is a wild concept to entertain. Now, I do want to make a quick but very important detour here before we wrap up, because Weisberger explicitly addresses this in the book. Whenever the topic of toxoplasmosis and mind control makes its way into mainstream news articles, public discourse almost immediately jumps to the crazy cat lady trope.
Yeah, that happens a lot.
People read a headline and assume that if you own cats, you are inevitably going to contract a brain parasite that makes you neurotically hoard felines, lose your grip on reality, and develop schizophrenia.
It is vital we address that misconception. As the author rigorously notes in the text, that stereotype is deeply flawed and scientifically irresponsible. The crazy cat lady trope is rooted far more heavily in systemic, historical misogyny and derogatory cultural portrayals of independent, single women than it is in pure, objective science.
Simply cohabitating with a domestic cat is not a one-way ticket to psychosis or hoarding behavior.
The actual mechanisms of a T. gondii infection and its subsequent subtle effects on human behavioral traits are incredibly complex. They are heavily dependent on a vast multitude of compounding factors, including an individual's unique genetic makeup, baseline immune health, and environmental stressors.
We must be careful not to let a nuanced biological reality be co-opted or simplified to reinforce a harmful, unscientific social trope.
The actual science of how a single-celled organism subtly nudges dopamine levels is fascinating enough without adding societal prejudice to the mix.
So, as we look back over everything we've uncovered today, where does this leave us? We've traveled from the humid understory of the rainforest where ants lock their jaws onto leaves, to the bodies of caterpillars violently guarding the cocoons of their attackers, to spiders building fortresses for wasps, to the possibility of human entrepreneurs being subtly nudged by latent cat parasites. When you stack all of these systems together, what is the big takeaway?
The core epiphany brings us directly back to the framing of our show, the crisis in perception. As human observers, we possess a natural, deeply ingrained cognitive bias to assume that the behavior we observe, whether it is a complex insect navigating a forest, a mammal reacting to a predator, or even our own daily decision-making, belongs entirely to the autonomous individual performing it.
We view organisms as closed, independent units.
But a comprehensive systems analysis of evolutionary parasitic manipulation shatters that illusion of total autonomy. It reveals that behavior is very often an emergent property. It is the result of intricate, unseen interactions between multiple competing organisms sharing the exact same biological system.
When we ignore that deeper ecological structure, an ant drowning itself in a pond or a mouse running eagerly toward a cat seems like irrational, inexplicable madness.
But when we zoom out and view that exact same behavior as a fierce evolutionary competition playing out on a molecular and neurochemical level, it makes perfect chilling sense.
It's a hidden, ancient war being fought continuously all around us and occasionally inside us, using weapons made of enzymes, viruses, and neurotransmitters. And that leaves me with a final, provocative thought for you to mull over as you go about the rest of your day. If microscopic organisms operating purely on blind evolutionary instinct have evolved the ability to so profoundly alter the physical bodies, the risk-taking behaviors, the deepest fears, and the daily routines of complex animals, and potentially even subtle traits within human beings, how many of our own daily preferences are truly our own?
That's the real question.
Think about your sudden mood swings, your inexplicable urges, your sudden bursts of energy or fatigue. How much of that is the autonomous you making a choice, and how much is a subtle chemical nudge from the vast, complex microbiome living inside your gut and your tissues? When you truly realize how interconnected and porous biological systems actually are, you have to ask yourself, where exactly does you end and your ecosystem begin?
Crisis in Perception examines how systems shape belief and how better tools help us see clearly.
This project is free and publicly accessible. If you'd like to support the time, tools, and research behind crisis and perception, you can become a supporter on Patreon.
For a shorter visual overview of this topic, see the mini explainer linked in the description.
If you enjoy this kind of systems-level analysis, please like, subscribe, and comment with books or topics you'd like us to explore next.